As the world's population ages, Alzheimer's disease, cardiovascular disease, diabetes, and various degenerative diseases are becoming more prevalent. Many of these diseases are associated with loss function of specific cell types.
Pluripotent stem cells are a specialized cell type that can generate almost all the different kinds of cell types in our body, such as the pancreatic beta cells or cardiomyocytes, so you can use new functional cells to replace the damaged or aged cells.
In the 1990s, the developmental biology technique somatic cell nuclear transfer became widely known thanks to the success story of Dolly the cloned sheep.
This technique can reverse the developmental clock of a mature cell by transferring the nucleus of a mature cell into enucleated egg cell. You can reprogram this mature cell to a stem cell state, and based on this, you can also regenerate tissues or even organs for future medical applications.
This landmark in the biosciences allowed Deng Hongkui, whose work originally focused on virology and immunology, to see the enormous potential of stem cells and regenerative medicine. In 2001, Deng Hongkui joined Peking University in Beijing and set up his own lab to pursue this research. In 2006, Japanese scientist Shinya Yamanaka and colleagues discovered that forced expression of four specific proteins, known as transcription factors, can convert fibroblasts into induced pluripotent stem cells, or iPSCs. However, the application of transcription factor-based reprogramming was limited due to the potential for random gene insertions and accidental activation of cancer-causing genes.
Reprogramming a cell is more like a systematic change to the cell's whole regulatory network. Because small molecules can easily enter cells, you can easily make different chemical combinations to treat different cell types with different dosages. So chemical approach is much more flexible than genetic approach. Moreover, this approach is transgene free and does not involve false expression of key cancer-related genes, such as c-Myc, making it potentially a safer approach.
In 2013, Deng Hongkui pioneered an approach using small molecules to convert fibroblasts into a type of iPSC known as chemically induced pluripotent stem cells, or CiPSCs. He showed that CiPSCs can be used to successfully produce living mice. His work also revealed the underlying molecular pathways that lead to CiPSC generation. The induction of pluripotent stem cells from somatic cells by chemical reprogramming involved three key sequential molecular events: erasing somatic cells' identity, generating an intermediate state, and establishing a pluripotency network. In Deng's mouse system, the first two events are completely simultaneously in the early stages. Initially, the combination of 5 small molecules, VC6TF, synergistically erases somatic cells' identity and induces the Zen-like state. At the same time, VC6TF induces the expression of SOL4 and SOX2, genes involved in maintaining the pluripotency and self-renewal of stem cells. Researchers found at the time that expression of the gene OCT4 acted as a master switch controlling the conversion of mouse somatic cells into pluripotent stem cells. Interestingly, OCT4 expression was inhibited by an epigenetic modification called methylation. Deng used this finding to add another small molecule in the later phase, DZNEP, to significantly reduce the methylation of OCT4, release the inhibition of OCT4 expression. In addition, VC6TF induced SOL4 and SOX2 expression, also stimulated OCT4 expression. OCT4, along with other pluripotency genes, formed a small gene regulatory network, collectively completing the last chemical reprogramming step to generate CiPSCs. Deng later went on to find that human CiPSCs induction involved the same 3 major steps as in mouse cells. However, it was more difficult to erase human somatic cells' identity and establish an intermediate state simultaneously.
Human somatic cells are more highly specialized functional cells. They're maintained with a cellular identity that is hard to be changed by chemical compounds due to a more stable epigenetic state.
Inspired by the regeneration mechanisms of injured somatic cells in more primitive animals such as axolotls, Deng Hongkui also succeeded in generating human CiPSCs and demonstrated that human CiPSC-derived pancreatic isolates can improve diabetes, showcasing the clinical potential of the technique. Deng Hongkui's seminal and transformative work has opened up a new route to cellular reprogramming that will have a broad and long-term impact on stem cell research and regenerative medicine. For his pioneering work on using small molecules to change cell fate and state, in particular to reprogram somatic cells into pluripotent stem cells, the 2024 Future Science Prize in Life Science goes to Deng Hongkui.